My name's James. If you don't know me, I'm the moderator for the talk tonight. And without further ado, we'll jump into our talk for this evening with Dr. Jordan Kuhnman. Did I pronounce your name correctly, Jordan?
Put my audio back on.
There we go.
Uniman, yeah,
Kuhneman. Thank you.
like the
Dr.
spice.
Jordan, there we go. So Dr. Jordan Kuhneman is a research associate at Cornell University working in the Danforth lab within the Department of Entomology. He earned his PhD in ecology and evolutionary biology from the University of Colorado Boulder, where he focused on conservation medicine and host-associated microbiomes under the mentorship of Dr. Valerie McKenzie. Following his PhD, he spent 4 years in Panama at the Smithsonian Tropical Research Institute studying plant and animal microbiomes. During this time, he led a large-scale project investigating the microbiome of bees across different life history strategies, seasonal patterns, and spatial distributions. Currently, Dr. Gunneman, thank you, runs Project GNBee, a research initiative dedicated to understanding why solitary ground nesting bees form aggregations in specific locations, identifying the factors that determine their long-term success, and working to protect nesting aggregations where they occur. Take it away, Jordan.
All right. Thank you so much for having me with you guys today and to share some of my research. So I've changed this talk a little bit. I put it together here on the road while I'm tracking down some ground nesting bee aggregations. But I wanted to share with you some results from a recent study that I worked on studying the microbiome of solitary bee brood cells. And I think that might be of interest to you guys. And because that's really shaped my experience working with bee aggregations and brought me into this project that I created called Project Ground Nesting Bee, which is geared and focused on locating bee aggregations, studying them, and conserving them. So this talk is going to be broken down into two parts. The first part is talking about those brood cell microbiomes. So that's on the microbial acidification of the bee brood cell stores, and in this convergent strategy that we have discovered. And then the second part is really on harnessing community science and bringing you guys together into this project in order to learn about bee aggregations and to protect them where they are. So the first part about the microbes, and the star of our show on microbes is this Diadasia rinconus, and this is desert bee from Arizona, but we have some nice photos and some art, so I wanted to share with you this brief little video of them going around. And this is really the starting point of this project was to locate where bees were forming aggregations, and that was required because I needed to get multiple bee brood cell pollen provisions from the same species. And the only way to do that without endless searching is to find an aggregation where you can dig out those brood cells. And the reason that I was interested in these brood cells and interested in the microbes in them um, is because we know that they provide a number of important biological functions, and that those microbes are provided for the offspring from the adult bee. They originate from the pollen and nectar because, as you know, they don't have overlapping generations, and so there's no sharing in microbial resources. But there's mounting evidence that suggests that these microbes are very important for the development of the bee and the health of the bee. And it turns out that a lot of bees cannot undergo metamorphosis and become adults without microbes in their diet. In fact, those microbes, in some cases, provide up to 20% of the nutrition for the bees, and sometimes we refer to those microbes in the pollen provisions as microbial meat. But microbes provide a number of other functions, including detoxifying toxins from flowers, from the pollen, preventing the pollen provision from spoiling underground. They can also liberate nutrients as they digest some of that pollen. They free up different proteins. And they can offer that direct source of nutrition that I mentioned in terms of the protein that's found within the bacteria and fungi themselves. And they can also prevent and help exclude other pathogens that might invade that resource. But despite all these critical roles that we're learning about in the bee development space, um, you know, very little is known for most solitary bees, especially ground nesting bees, about their microbial associations. And so we don't know how ubiquitous these different um, functions of microbes in the brood cells are. And so that's what this was meant to help uncover. So here's a site that came out a little blurry, but here in New York. And I just wanted to sort of point out like where we would go to find these aggregations. And in this little tour on the bee brood cell microbiomes, you really have to dig down in order if you want to study them. And so one way we do that is by for a species that we're unfamiliar with, is we put a little talcum down the hole, we blow it in there, and we excavate little by little in order to get down to the site where the pollen provision is or the larvae is developing. And then we can move laterally and find fresh, clean pollen provisions in order to study those microbes. So at that point, you're moving into a more of a sterile technique once you know what you're looking for and how deep you have to go. In other cases where bees made very deep nests, sometimes I would burrow into the side of the mountain in order to get to those brood cells. So here's me digging away in the highlands in Panama. And I was able to do so in that place because this bee, Crawford apis, forms a very enclosed brood cell here with a cellophane bag and a little venting cap. And so we could access the contents once we basically extracted out these little nuggets of biodiversity from the ground. And now here's a little collection of what these different brood cells look like for 12 different tribes that we were able to study. So actually, this is a representation of 12 tribes, but we collected over 340 brood cells from 6 bee families, so 18 tribes in total and 39 species. And the goal was to just get a kind of wide collection to look at sort of what's going on in bees and these different strategies that they may have. And if we look at the phylogeny of those bees, we can see they're pretty diverse. Again, those 6 families, most of which were in the Apidae, but some other bee families there were included. Most of these bees in this study were ground nesting, but still we had some cavity nesting, stem nesting, and wood nesting. And we had a fair, fairly much an even separation between specialists and generalists. So we had about 30 pollen nectar generalists and 16 pollen nectar specialists, and 3 specialists were oil collecting bees. So as you zoom into those bee brood cells, you can uncover that biodiversity that's within them. And here we analyze the bacterial and fungal communities from those brood cells using 16S and ITS amplicon sequencing. And we're only going to talk about some of the results from the 16S, which really was the more interesting part of the story. In this plot that I'm showing you here, I've organized the bee species that we had. Those are along the x-axis into their tribes, with the sort of remainder of them over here in tribe other. And what we can see is that bacterial richness within these provisions were variable across species, but sometimes consistent within tribes. So bees in the tribe Centrodini had the highest brood cell diversity with respect to their bacteria. And those were consistently higher than everything else. And those are our oil specialists, include our oil specialists, whereas another group that was higher than most was the Osmini, and these were stem nesting. So next, we can see some other groups that had very low microbial diversity compared to other groups of bees, such as the Caupilocanini. Those were the diphoglossines, and we'll talk about them a little bit more. The Amphorini had consistently low brood cell diversity, and also Nomine had low brood cell diversity. Whereas a few other tribes that were in this study had mixed results. So some species had low diversity, some had high. And certainly those groups also weren't further investigation. Couldn't sample all the bees that we would hope to, but we're working in that direction. When we consider the composition of the microbes within those brood cells, again, with the species on the x-axis organized by tribes, and the relative abundance of the 13 most dominant bacterial families, we see some consistencies, see some patterns emerge. The first of which is that the Lactobacilliaceae is the dominant bacterial family within those brood cells, followed by Leoconciaceae and Acetobacteriaceae, were interesting in the context of lactobacillus because those are all acid producing bacteria. Um, and I put a little note here on the bottom that's covered up here, but it's showing the pKa of lactic acid versus acetic acid. So lactic acid is a stronger acid than acetic acid, which is produced by those Acetobacteriaceae and the Leoconstiaceae. But both could be quite significant in acidifying a brood cell. And another way to view that brood cell diversity with respect to those acid-producing bacterial families is to look at their abundance, summarized by bee tribes. So in the Acetobacteriaceae, only a few tribes, such as the Aglochlorinae and the Euglossinae, had, significant amounts, whereas the Lactobacilliaceae was found within quite a few of the bee tribes, and noticeably absent in things like Anthenodini and Anthophorini, some of these other ones, such as Osmini. So its absence is actually quite interesting. Whereas the Leoconostiaceae was really only found in the Amphorini. And so it kind of starts to tell this story about specialization and how certain bee groups are utilizing certain microbes that are acidifying those pollen provisions. But that can be explored in much greater detail when you look at the strain levels, or at least here showing the different sequences that are from those acid families in a phylogeny, and overlaying that phylogeny with the bee phylogeny, we do see that they are co-diversified together. It's a weak signal, but it is significant. So there are certain strains that are associated with certain bees. And that's more obvious when you consider just the different strains of lactobacillus, and even though the strains of lactobacillus that do not have species named yet, and how those occur in very specific tribes. And one of those specific tribes that I mentioned earlier, or at least in the the Diphoglossine family, this is the Coppolocanini. We studied this group more in depth with 3 different species, and using a number of more advanced techniques to get at the brood cell diversity. And these guys form sort of unusually fermented brood cells. So you could even observe this in some of the brood cells where they smell very pungent, and they, in some cases, were even bubbling. And one of those bees in this study was the Tiloglossa arizonensis. So this was work done with a colleague, Toby Hammer. And this awesome graphic was actually 3D rendered, but shows the major pollen source and the major nectar source. So nectar being agave giving nectar, and pollen coming from solanum. And down here in the bottom right corner, you can see the brood cells in the earth, and they're very liquidy provisions. And when we studied the microbial diversity, or at least the bacterial diversity from those larval provisions, we also studied individual bees, so developing larvae, mature larvae, the adult gut, and even some of the nectar from that plant. And what we see is that these bees, as well as other samples within this group, are near monocultures of lactobacillus. This is an apilactobacillus that happens to be fairly widespread, and even found in other bees, but was absent almost entirely from the nectar source. So it leads us to sort of this conclusions that it must be the nesting biology of these bees that allow for this fermentation and the brewing of this lactobacillus within those brood cells. And certain attributes of bees may facilitate some of these microbe interactions. So them being underground and having more stable brood cells in terms with regard to temperature, having a high water content and suitable sugars like fructose. The fact that they're enclosed in cellophane bags in this case may provide some sort of protection from invasive microbes, although we can talk about that more later. Um, and even these brood cells and their linings seem to allow for venting, which is an incredible adaptation if you're having a highly fermented pollen provision as your food resources while developing. So we wanted to show you a little bit more about this strategy that we observed and how it seems to be a convergent strategy. So when we started looking, testing the data and just seeing, how whether or not we could generate good data, we ran 6 different species from bees that were very different in their biology. And what we saw was that half these bees had a restricted microbiome, all dominated by lactobacillus, and half of them did not. So this is a weighted unifrac, and so that not only did they have these lactobacillus, but they had lots of them. So it really separates their microbial communities from other species. And so in this plot, each circle here is a brood cell from a bee species, and it represents the entire community of the microbes within that brood cell. And so you're looking at a dissimilarity matrix, and so samples that are close together are more close together within their communities. So they have the same microbes in those communities, and if they're farther apart, they have differing microbe communities. And when you look at this again, but with respect to unweighted unifrac, it separates out in sort of another way. And it tells us a little bit about that, being a plant specialist doesn't necessarily um, make you unique from generalists, but rather the diversity in those provisions, the richness tells you a lot of the story. And some plant specialists, bees, have very highly diverse microbial communities in their brood cells, and others have very restricted brood cells, whereas the generalists were more in the middle. So that was kind of interesting. And if we summarize really what we saw, and if we're looking at the community composition, across the different levels of the bee taxonomy, so we see sort of increasing um, so we see that, as you go from family to the tribe to genus to species, that more of the variation is explained. And so the species effects are really matter. So there's these very important differences within the species. Whereas some of these other functional traits, such as being a generalist or a specialist, or your nest preference, or even your life history, whether you're communal or really solitary or facultatively social, didn't matter nearly as much. So I thought that was pretty interesting, and I wanted to share those results with you guys before we move on into Project GNBee. Um, and some of the things that are left to, explore here, there's quite a few of them, but, we really need to look into some of these bee tribes in greater depth. We should start to consider the bee brood cell microbiomes from species occurring in the same locations to try to remove some of the spatial variability. Um, it'd be great to sample for water content and pH and see how those uh, are reflected in the brood cell microbiomes. And it'd also be great to sort of assess the nutrient acquisition from bees of different brood cell microbial enterotypes. So if you were dominated by lactobacillus, how much are nutrients are you acquiring from lactobacillus or from the pollen provision versus brood cells that have very high diversity with lots of different microbes? Are you getting more nutrients from uh, the bacteria in those communities than other samples? So with that, I would like to move on to the second part of the talk, which is which is bringing into you guys into the conversation of how we can study bee aggregations and conserve them. And let me see how I'm doing on time. If somebody could tell me, that would be great. But this is really the big overview of this project. So Project GNBee is a community science project to study and protect ground nesting bee aggregations and to support their vital pollination services. So we've got one arm that's really in the research and locating bee aggregations and researching them. Another arm that's working to try to protect bees where they are. And then we want to take both these parts of this information that we're gathering and see how we can apply it to manage bees in the landscape in ways that are beneficial for humans and for ecosystems. This project is a community science project. So it really builds off of efforts done at the local level and you guys that are participating in it. Um, and when you find the project on iNaturalist, you can, this is the banner that you'll be shown, so you know you got to the right place. And what we're asking you to do is look for bees hovering around the ground, generally in spring, all the way through late summer, and to take photos of those bees and those locations and upload them to the project. At current, we have over 5,600 observations from 380 species, and 2,200 folks have added observations to the project. And it's really important to acknowledge all the folks that are identifying those photos on iNaturalist that make this all possible. And also the little Hall of Fame here for those that have made the most observations and observed the most species and so forth. So we recognize them as well. When you find the project, if you can, please consider joining. It really helps us know who to reach out to and who's engaged on the project so we can gain more information about the sites that are being observed. And on iNaturalist, new sites are have these little icons on them so that you can sort of see if anything new has popped up in your area. So within Washington, we currently have 209 observations. So um, maybe about that many aggregations, but sometimes we encourage repeat observations from the same aggregation. And this shows you the species that have the most observations um, in Washington state. And the goal here is to be able to study bees at different scales. So study bees at local, regional, and national scales, and learn why they nest where they do and what allows them to be successful through time. So I'm going to take you through a little project that happened recently. This is showing an area around Cornell, and one of our lab members was walking home after work and came across a bee aggregation in a cemetery, and that prompted our first in-depth study of a local bee aggregation. This species happens to be Andrena regularis. Um, so this was observed in April 2023, and I'll show you a little bit of what that aggregation looked like and let this play for a second. Um, but basically, it's a very robust aggregation site. I threw that little uh, flag in there for perspective. But we were also marking areas where we were going to dig in and see what was going on underground. Well, let's see if I can move on. So this aggregation occurred in the cemetery, and I point this out and wanted to share that because we think cemeteries are very important for bee biology, and they can be very interesting opportunities for bee conservation as well. Um, and that's great because cemeteries are sort of overlooked largely nowadays. I don't see anybody in them when I go, but I'm in them a lot looking for bees. And so what we did in this site was put out a number of emergence traps across the parts of the aggregation where we saw bees nesting. And with one of the emergence traps, and the highest densities, we found up to 2,000 bees that emerged, and that was very shocking. So it really changed my perspective on how many bees can exist in a square meter if you're in the right spot. And so this part of the project was led by a student, Steve Hoge, in the Danforth lab. And putting together those estimates of the number of bees that emerged and the area of the aggregation, we estimate that 5.5 million bees emerged, and that would be happening every season. What's interesting about this is that these are great apple pollinators, and they emerged during the time of the apple bloom, and they are located about half a kilometer from the apple orchard. And so, for a long time, we were seeing these bees in the orchard, but we didn't know where they were nesting until more recently. So it's a really nice direct opportunity to link the biology of a site to pollination services that are providing food. And we also learned that from this studying of from emergence traps that the dominant parasite was Nomata luteolioides. So I now want to expand on that, and so basically look for ways of taking this in-depth study of one aggregation, but expanding it out again. This would be more of a regional level, um, and taking what we can learn from these different sites and making a description of what Andrena regularis needs to form a nesting aggregation. And all these sites have sort of varying levels of success in terms of their size, so we can start to get a framework for what allows bees to be successful where they are. And one way that we're doing this is pulling in a lot of spatial data. Most of this data is available from USGS. So we have the soil survey, so we can get at soil texture um, based on those uh, landscape surveys and pull that data in for sites that we haven't visited. Landfire has a lot of great data, so, canopy cover and aspect and slope, and um, also the USDA crop data layer has been fantastic. So we're starting to pull in different types of landscape data with regards to the plant life around these different aggregations. And that is moving us closer and closer to understanding why bees nest where they do. Um, I'll walk us through another species here. This is one that I'm currently working on. So I am actually on the road in a hotel, driving to these different aggregations in order to take some measurements and collect some observational data from the sites where they're reported, and to figure out which sites would be good for continued work. And so I will go to a site and try to look at, take in the surroundings of that site, work on getting a sense for how many bees are nesting in that area, and a number of different observational data that we can talk about more in detail. So this is me from earlier this morning, showing up at a site with my net and my shovel and some collecting tubes, headed out to study this aggregation. And we can take those points and use, like I said, some of these databases in order to get uh, more information about what they're nesting in and why they may be there. I'm working on validating that with actual soil samples, and so that's where we are with the project now. But these bees like sassafras, and we can also map where sassafras is in the landscape, and we can see that these bees are largely falling in the areas where that soil layer occurs. But I mentioned that I'm collecting physical data from those sites as well. So I'll collect about 3 cups of soil from each site that I can visit, digging down to about the depth of the brood cells, and then running those soil samples for texture analysis and for nutrient analysis, so we can start to get real numbers associated with things like the percent sandstone clay and the organic matter, as well as a number of other cofactors and metals and so forth that might be in that soil. So trying to figure out what allows them, why they choose to nest where they do. And then pull that data in and explore it in different ways. So, in looking at the categorical data, trying to figure out, what is the pH that certain bee families or bee genus or bee species can tolerate. How is pH linked with other attributes such as like the sand or silt? Um, you can pull all that sand, silt, clay into these texture triangles and start to look at where what kind of soil they're nesting in, and does that group with different bees as well, and estimating basically the distributions around the means for those values in order to create a profile for those bees. And so we're currently working on bringing that data together to do some modeling and basically some multivariate analysis that will incorporate attributes of the soil, attributes of the site, um, and other attributes in the landscape, such as the crop layers. So, if you would like to be a part of this project and like to contribute and towards understanding why bees nest where they do, here is a QR code. So we would gladly take observations that you would make and follow up with you about those observations. Here's a map of observations that are currently exist in Washington. And so one way to kind of get going with this is to consider visiting an aggregation that's close to you. It's a good way to sort of learn what you're looking for. It's a good way to share um, bee diversity and bee biology with other people that haven't seen it. But those of you in this group, which is probably most that know what you're looking for already, feel free to just wander about your lives, and hopefully, if you come across an aggregation site, please share it with the project. One thing that I love about visiting these aggregations is that most of the bees that we're working with will nest in the same location year after year. So they're reliable, and you can look at when a bee was active in the year prior and use that as a guide in order to go and find them active again, hopefully, when you show up. And these bees are often, let's just say, slightly harassed by other bee parasites in their environment. And so it's fantastic to make observations of those as well. So we can start to really bring together the bees and their parasites and do so across the landscape. So they're not just one-off observations, but um, we can start to see how strict are these associations. So when you're making your observations, we ask you for a couple things. One, try to get us the best photo you can of the bee itself, so we can get an idea at least what genus it belongs to, but sometimes it's a species if we know the bee well. We'd like photos that are provide a little bit of context to the area. So what is the environment look like, where the nesting is occurring, and if you can also provide us with a photo from the sky of that location so we can get a sense of how much canopy cover there is, and there's notes that we would love to have, such as what maybe is the slope and what is the aspect, um, and very importantly is how many bees do you observe nesting. Give us an estimate, and if it is a site that is robust enough to warrant further exploration, then you can come back to those sites, and you or maybe somebody who that might be willing to just map out the boundaries of where you're observing those bees, and if possible, within some meters from the center of the aggregation, we can count the number of nest entrances, and by getting just those two metrics, we can really get a very good estimate of the number of bees. And if you have an aggregation that you are really close to or become very close to, it would be wonderful to try to do this, at least trying to estimate the density and the abundance, if even if the bounds remain the same, year over year, so we can get a sense of how long term are these aggregations, or is there anything negatively affecting them, and start to, think about protecting aggregations in the landscape and how best to do that. So this is one way that I've done, was just putting in a meter by meter quadrant, and basically summing up the number of nest entrances that I observe and multiplying that by the area of the aggregation. We also have people, and I've come across several of these as well, where you have numerous species nesting in the same area, but they sometimes nest in sort of restricted areas within that space. It's really quite amazing if you consider all the space that a bee could nest, that it's only willing to nest on this sloping face, in front of this building, and they have to share it with other species. And so I find this just really fascinating, and so it tells us very clearly that they have strict requirements for nesting, and we just want to figure out what those are. And here's a little shout out to Kevin, who also provided us some great information on a nesting aggregation in his area, and provide more information on the history of the area, which is helpful when making the case and the argument for what allowed those bees to be successful, how long have they been in the environment, and what needs to be done to protect them where they are. So I'm very interested in some of these very large aggregations on trying to get a real estimate of the age of the population. And sometimes the way to do that is from historical records as to, when was this place logged for the first time, for example, or when did somebody report bees in some survey. So that kind of information can be really helpful just to give us a framework to work from. So thank you for that. And if you find one of these aggregations and it happens to be substantial and it fits within the priority groups that we're working on, which are largely Antrina, Calides, and Melisodes, but some other genera as well. This is sort of a first pass with bees that form very large aggregations. We've been sending out envelopes to folks to get them to collect a few cups of soil, and they're prepaid that you can send them back, and we'll have them analyzed and they'll be incorporated in the study, and we can share the data with you so that you get to learn as to what the bees are nesting in, and maybe use that information the way we would to try to mimic or create nesting resources that are similar to what they're using it. And so looking forward, this is really where this is going, is sort of integrating all these different attributes of mapping aggregations and modeling their nesting requirements, collecting soil samples and screening bees in those soils for pesticides, identifying, priority conservation sites. We've been doing a little bit with testing whether we could relocate bees from one aggregation site to nesting substrate where they're not. Of course, photography is very important in this project, collaborating with orchards so we can have a direct um, sort of applied management from our work. And this is what we learn, and how we learn is through employing these emergence traps and identifying bees as best we can. And the hope is to, have all this information feed into policy to help protect bees, and to just keep bees around in the environment, and to enrich our lives with their presence. And so we're very grateful to them, and we're very grateful to everybody who has participated in this project, and to you guys for taking the time to listen to it, and hopefully for getting out there to observe your local aggregations and provide some additional data. So I wanted to thank you for that. And here are some links. If you want, the link gnbee.org is where I provide some basic information about the project, and that can get you to the INAT project as well, and how to make observations. And we put some highlights on there for folks to see. So with that, I thank a number of collaborators on the Bee Microbiome project from various universities around the country, and for the army of undergraduates and folks in the Danforth lab that are engaged in this project as well. So I really wanted to thank you as well. So that's
Great.
it.
Thank you so much for that. That was an awesome presentation, Jordan. That was really fascinating.